Hello, everyone. My name is Mya Thu, founder and CEO of VisiCELL, GPS for cell therapy. I have more than 15 years of directly related experience in this space and have previously taken the technology from lab to market and contributed to successful exits of 2 biotech startups. Today, I'll be showing you how VisiCELL is providing a proprietary, rapid, and actionable non-invasive platform technology for proactive management of translational IND-enabling studies and its follow-on clinical studies in this multi-billion-dollar cell therapy industry for an accelerated time to market. The STEM and immune cell therapy is in its infancy with many unknowns. However, the industry is rapidly growing with the current success of genetically engineered immune cell products. For example, Chimera that saves patients like Emily, who has less than 20% chance of survival due to aggressive leukemia. There are 5 products, immune cell products in the market, and the companies are rushing to win this market opportunity with their promising cell-based therapies in both oncology and regenerative medicine applications. However, the major challenge in this space is we currently do not have an easy and safe way to visualize the cell products in continuum from the animal models to the human patients during the product development. To understand cell biodistribution, engraftment of these cell products, and the toxicity and safety associated with such activities. The scientists are dealing with the black box, which has serious negative consequences, such as the extended development timeline and the delayed product failures while taking these promising technology to the market for approval. After speaking with more than 50 multi-stakeholders, including translational heads and investigators from the public and private sector, we learned that the following imaging insights can directly or potentially benefit what they are currently working on. For example, biodistribution information for FDA required safety, on target off-target toxicity for tox management, and cell type specific quantitative infiltration at the disease site for dosing and treatment strategy. And last but not the least, engraftment and its associated response or disease burden for understanding treatment efficacy. The current standard of care or practice to monitor the cell therapy products is PCR or histopathology and biopsy, as shown on the left quadrant. PCR and histopathology or biopsy are inherently labor-intensive and provide single time point measures with extensive sampling errors associated with them, and simply does not solve the challenges when it comes to looking for the cells in the body, like a needle in the haystack. PET imaging, as shown on the right, allows for whole body imaging, however, uses radiation and radioisotopes, and the equipment and the radioisotopes required are not easily accessible. VisiCELL employs standard MRI and optical imaging for easy accessibility while providing whole body imaging in 3D settings for an unprecedented insights, enabling a feedback loop for the product development. There are 2 main methods for labeling of the cell product while considering the methodology. Direct labeling, where the labeling agent or dyes are introduced directly into the cells before injection into the subject, or indirect labeling, where the reporter gene is inserted into the cells using genetic engineering technologies. The advantages of direct labeling are straightforward clinical translation, and it is best for short-term biodistribution study, for example, days or weeks. The downside is label dilution issues. The indirect labeling is suitable for long-term tracking, for example, 1, 2, or 3 years and cell viability studies. However, it involves complex manufacturing due to the requirement of additional genetic engineering. The trade-off here is the endogenous contrast with the immunogenicity. After getting the feedback from our target customers, we created a solution that locates the cells in the subject and is simple and easy to use. Our patent pending solution labeled the cell product with biodegradable nanoparticles before injection to the patients and uses standard MRI and optical equipment available in most facilities to obtain biodistribution and safety information. Supporting our solution is a fully developed and well-validated core technology that was published in high-impact journal Nature Medicine, where I was the first author. The innovation here is its clinical applicability and versatility of these self-assembling nanoparticles with a proprietary formulation using individually FDA-approved products. We are looking at here these safe and biodegradable nanoparticles in the label cells, which were internalized and encapsulated in the endosomes, as seen by TEM in T cells, MSCs, and MSCs. There is no significant toxicity detected in these label cells in comparison to unlabeled control, as assessed by 3 different assays: left, apoptotic assay, middle, mitochondria activity assay, and the right panel, reactive oxidative species assay in 8 different cell types. Let's look at a few use cases where different cell types and disease models were deployed using our core technology. What we are looking at here is MRI visualization of labeled MSC in a transgenic mouse model of Alzheimer's for up to 2 weeks. We are here looking at the labeled MSC in Osteoarthritic Model as hypointense MRI signal as indicated by the red arrow, and for up to 4 weeks. These images demonstrate how our core technology is used for strategic decision-making in selection of the dosage of cells and route of administration in an IND-enabling studies of genetically engineered neurostem cells in glioblastoma models. This cell tracking technology was further amended in an existing IND and included in a phase 1 clinical trials for observing cell product biodistribution. The left panel shows the extent of dose-dependent cell product homing within the tumor, while the right panel shows the cell product biodistribution at the tumor site via IV administration as seen by MRI. Our core technology in action that shows tumor target engagement with these labels, immune cells, natural killer cells in this case, at the tumor site within 2 days. The paper also shows the number of cells infiltrating at the tumor site directly correlated to the reduction in tumor volume, which can be a secondary outcome measure. We have since developed next generation VMI track nanoparticles to further improve upon our core technology, the standard HPF. for very difficult to label cells such as MSCs. The graph shows an increased viability profile of our patent-pending VMI-track nanoparticles formulation in comparison to the standard HPF. The labeling efficiency and iron content per cell was also tremendously increased by several folds. What I would like to highlight here is the users can label their cells product with our VMI drug nanoparticle formulation in serum containing media, which provides greater versatility and not requiring substantial changes in manufacturing processes, such as stirring the cells. Taking a step further, we developed next generation VMI drug V3.0 nanoparticles for very sensitive, very small immune cell products such as CAR-T or CAR-NK cells. The graphs show astoundingly better viability profile of VMI drug V3.0 formulation in comparison to standard HPF and 100% intracellular labeled cell population in both cell types. In response to our customers' feedback and the need for continuum between animal models and human patients, we also developed a dual MRI optical agent in a single nanocomplex. And as demonstrated here, can be seen in fluorescent and with the iron sensitive Prussian blue staining, ready for the MR optical dual imaging. This is our product pipeline with each product made for the specific stem or immune cell type. VMI track dual that allows for dual imaging is developed and customized for specific cell product with our partners or customers. Behind VisiCELL is a strong team with more than combined 100 years of experience in this space, including physician stakeholders such as radiologists and cell therapy implantation surgeons as co-founders, getting us ready to take from preclinical to the clinical arena as the technology moves forward and advances. It doesn't matter where your technology development stage is with your cell therapy product. Real-time cell location information can add values and economic benefits to it. For example, selection of lead candidate or route of administration in the subject in very early preclinical research setting, or providing the FDA required biodistribution information in IND enabling study and further supporting tox efficacy animal study. The information can also guide analysis of efficacy data in low power early phase clinical trials. All together with enough amount of data, these may all provide imaging marker capability as a companion therapeutics in the clinical setting. All of these contribute to accelerated product development from preclinical to clinical setting and help with rapid decision making during the product development and clinical development, and ultimately shorten time to market and improve patient care. We are looking for partnership opportunities at this time, and we are open to collaboration for evidence-based use cases and strategic co-development partnerships that could mutually benefit while meeting your unmet needs. Please contact me at mya@visicellmedical.com for further discussion, and I'm very much looking forward to talking with you soon. Thank you very much.